Good afternoon. Welcome to this IMSc Diamond Jubilee Colloquium. As you know, this talk series has been organized by my colleague Professor Sayantan Sharma, celebrating the 60 years of IMSc this year. Today it's a great pleasure to introduce our today's speaker, Professor Hema Rajaram. Briefly, Professor Hema Rajaram did her bachelor's in chemistry from Delhi University, subsequently master's in biotechnology from JNU in Delhi. Subsequently, she joined the BARC training school in 1994, and she has been in the bioscience group subsequently then. She did her PhD from BARC as well, and she got her PhD working on heat stress response in Anabina. Since 2012, she has been heading the Cyanobacterial Stress Biology and Biotechnology Division in BARC within the Bioscience Group. Also, since 2014, she is the Dean Academic Life Sciences of BARC. Her general areas of interest are DNA repair and biofill formation and bioremediation. It's a great pleasure to have you, ma'am, today. Give this talk. Over to you.
Yeah, please. Thank you for a nice introduction, Dr. Samar. So I'll be taking you through a lot more biology than the computational aspects of it, which is what you people would be used to it. And I'll be taking you to very ancient times, because I'm going to talk about one of the most ancient bacteria, that is the Cyanobacteria, and how it had combated environmental stresses, and whether there is any link between the environmental stresses as we know today in the mesophilic or other organisms and what existed in Cyanobacteria. So if I'm saying this Cyanobacteria has been on Earth since 2.8 billion years ago. So you know that it would have faced several stresses, some of which I have listed here, and the most important of these are the temperature stress, ionizing radiation, desiccation, oxidative stress, and heavy metal, because these were the things which were around for that longer time. So how did this organism actually respond to these environmental stresses, which would have been even simultaneous? And this would be through complex regulatory networks, through multiplicity of genes, and also cell surface changes. So I'll take you through different stress responses in this thing. And before I go into it, I would like to introduce the Cyanobacterium, which I'll be talking about. And this is Nostoc species strain PCC7120. Okay. So this organism you see is actually, it looks like multicellular, but it is actually filamentous. Now among Cyanobacteria you have two kinds. You have unicellular or you have filamentous, okay? And all of them are photosynthetic. There is a temporal separation. That means the photosynthesis occurs during the daytime and nitrogen fixation occurs in the night. But the filamentous ones, which are nitrogen fixing, like this Nostoc or Anabina 1720, we have specialized cells which are marked here, called the heterocysts. So these heterocysts actually develop from these vegetative cells. Vegetative cells are the ones which do the photosynthesis. So what happens is, they terminally differentiate, lose the photosystem here, and they become thick-walled so that oxygen doesn't permeate, and there is no evolution of oxygen because PS2 is not there. And thirdly, there is something called Nif Gene Rearrangement which takes place. So Nif gene are the genes which are coding for the nitrogenase complex. So this is, we have heard of the world of introns in terms of eukaryotes, but this prokaryote had it long time back. So what it does is, this Nif gene rearrangement takes place only when the heterocyst is formed. So once the heterocyst is formed, the intron, which codes for the enzyme to remove the intron, removes it. As a result, you have the continuous Nif gene and you have the product for synthesis of the protein. So this ensures that it does not waste the waste its energy in synthesizing this protein, which cannot function if oxygen is present in those cells. So now we know that intron splicing is not something which is only in eukaryotes, but even prokaryotes had it to begin with. So, and this was the second cyanobacteria to be sequenced, and that was way back in 2001. The very first one was the unicellular Synechocystis PCC6803. Now this has 1 chromosome and 6 megaplasmids and total of 7.2 MB genome, which is very huge for a microorganism. And in this, there are over 6,000 protein coding genes. 50% of these genes are either hypothetical or unknown function. That means these are not similar to the known proteins in the bacteria where these functions have been studied. And 83 are RNA genes in this. So what I will do is I'll just take you through different stress responses and what are the new things we found and how it could be related to what we know in modern day science today. So the very first stress response which was studied was heat stress response. So as we see here, this is in Anabina L31, another cyanobacteria. The response was very similar to what we see in normal bacteria. This is a radioactive gel and you see a time-dependent synthesis of proteins. So what was interesting was that we found 2 GROEL or HSP60 proteins, the 61 kilodalton which we called as CPN60 and 59 GROEL. Now it is not that this was something new for prokaryotes. There were a few other nitrogen fixing bacteria where multiple GROEL genes were found, but these were found to be regulated under nitrogen fixing conditions, and you could easily delete these genes and it did not affect either the stress response or the normal viability. But what we found was that we could not delete these genes. That means both these genes were required in the cell even under normal growth conditions, and they could not complement each other. But the other interesting feature which we found was this CPN60 protein, which you see here, under nitrate supplemented, that is non-nitrogen fixing conditions. You see it under control conditions, but the moment you give heat stress, it disappears. So it is a misnomer, that is, it's a heat shock protein which actually disappears during heat shock. So that was something which was interesting, but we were not able to find out why that happens though. So then since we were not able to obtain the mutants and we wanted to know the physiological relevance of these proteins in terms of whether they're involved in different physiological processes, so we overexpressed these proteins and looked at different physiological parameters such as photosynthesis, nitrogen fixation, and nitrate reductase abilities under nitrogen fixing and nitrogen supplemented conditions. And what we were able to find out was that if you overexpress either of the 2 HSP60 proteins, it actually helped in photosynthesis. But between the 2, the GROEL was the one, the 59 kilodalton GROEL, which actually supported nitrogenase activity, while the CPN60 was the one which supported the nitrate reductase activity. So this indicated that they do have distinct functions. We did not try to find out what are the other proteins which are specifically folded only by one of these 2. But what was interesting, which we found during our biochemical studies, was, see, this GROES gene is actually part of a operon, that is GROESL operon, okay? And we found that the GROEL protein does not require the GROES for folding, but it may be required by the CPN60, which kind of explained as to why we could not do with deletion of either of the 2 genes. So then the next question was, we wanted to know is how are these genes regulated, because this was completely unknown at that time in cyanobacteria. But in other organisms it was very well known. The very first one where it was found was in case of E. coli. In 1990s, they found that positive regulation was by sigma 32, that is RPOs. That was when they started talking about different sigma factors involved in stress regulation. But at around the same time, in the gram-positive bacillus, they found a negative regulatory element called the HRCA by Wolfgang Schumann's group. And then in 2000, another positive regulatory element, that is RPOE, was found in E. coli, and mRNA-based regulation called the ROS element was found in Bradyrhizobium. And since we already had the genome, There we could not find a sigma 32 like protein in terms of homology search, but we found a HRCA like protein in our organism. So which kind of indicated that if there is HRCA element, then HRCA protein, then it should have the SERS element. SERS element is nothing but, which I'm showing here, it's a 9 base pair inverted repeat element to which the HRCA dimer binds. So we also were, since we had found one element, but we wanted to see if there are other elements of regulation also. So upstream of the grow ESL, when we looked in, we found several inverted and direct repeats, out of which we found that something which we call as the H box here, which is an inverted repeat element, we found that it was a cis regulatory element which was involved in heat stress response. So under heat, the cis element opened up and allowed for higher transcription. And the K box was, at the same time when we were looking at it, it was reported in Synechocystis as being required for stimulation under light and heat conditions. But we found that it was only required for stimulation under light conditions, when light was being provided. So basically we were able to identify 3 elements for the grow ESL gene, but only the HRCA SERS element in case of the CPN60 gene, where other elements we could not find out. We did not try to look for the presence of H box upstream of other HSP60 genes, but we found that this H box is present across the filamentous Cyanobacteria. So it's a very well conserved mode of regulation. In addition to this, what we know is that what was found in case of Anabina were 12 sigma factors, out of which these 3 were found to regulate some of the known HSP60 proteins. Though they were shown under desiccation stress, but they were the HSP proteins. Also, histidine kinase were also shown to be involved in heat shock response, one of them being equivalent to the HIC33, which was identified for Synechocystis. So what we see here is that in Cyanobacteria, we had a plethora of regulators, including the sigma factors, the SERS element, and certain mRNA-based elements like the H box and histidine kinases. But when there was an evolution, we see the sigma factors was something which was restricted to the gram-negative bacteria. The SERS element was for the gram-positive, while the histidine kinases is now shown only in case of eukaryotes. So possibly there was a forebringer of all these elements was there in Cyanobacteria, but a need-dependent evolution took place and some of these elements were lost. Then the most important of these responses is radiation stress response, and why I'm saying that is because the radiation levels were very high when Cyanobacteria came into existence. So it would have had to develop a very good system to repair its DNA, because that is one thing which gets degraded quite this thing. But when you talk about a bacterium which is resistant to radiation, it is usually we talk about is Deinococcus, which is what people have studied, but not so much on Cyanobacteria. But here you will see that I have given it up to 15 kilogray of gamma radiation, and you see the degradation of DNA, but still most of it is over 100 kb. So it doesn't really get degraded, unlike where E. coli, even with 1 kilogray, you will get less than 20 kilobases of DNA. Okay, and when we allow it to repair, which is at say here we have given 6 kilogray, which is the LD50 dose, within 2 days it is repaired. Now 2 days you might think is very high, but then this organism doubles once in a day. So for them 2 days is just 2 generations. So within 2 generations it has repaired its DNA, which shows that the repair mechanism has to be very efficient. But sadly, because of the non-availability of genetic manipulation techniques for very long, which was developed only in late 1990s, as late as 1997 and 1998, that this kind of work was not being taken up very effectively. And many of our proteins don't fold very well in E. coli, which is the workhorse. So on basis of only what is known in other bacteria, we are trying to predict what is happening in Cyanobacteria. So if you look at in gamma radiation, what is the main damage which occurs is the double strand breaks. Okay, so what are the different Dsb repair pathways which are available? Are homologous recombination, are non-homologous end joining, or sequence dependent strand annealing pathway. So in this, the homologous recombination, you have 2 pathways. One is RecBCD and RecF. Now in this particular organism, the reg B is truncated, reg C is absent, while reg D is present. So the reg BCD pathway cannot be functional, whereas other genes of the genes of reg F pathway are available. Same way, if we come for non-homologous end joining, we do not have the Ku proteins. So without the Ku proteins, the NHEJ pathway will be ineffective. It cannot take place. So this is also ruled out. But there is an alternate NHEJ pathway called the microhomology mediated end joining. So this requires a lot of repeat elements, and Cyanobacteria is replete with such repeat elements, which are say 5 to 7 bases long. So this is a possible pathway. Similarly, the proteins which are required for sequence dependent strand annealing or extended synthesis dependent strand annealing, those proteins are also present. So these two pathways could also be present in this organism. So what we feel is that basically in this organism, it is a synthesis dependent repair which takes place, and what helps it is because of its long generation time, which allows the synthesis dependent pathway to take place. So based on that, we were just able to predict the different proteins which could be involved in these pathways. So when we talk about what are the proteins which are required, see, the very first thing which comes into my mind will be something which is going to protect the single stranded DNA which is present, which is generated whenever there are double strand breaks, because there will be nucleus which will try to eat it up. So you need an effective single stranded DNA binding protein. But what was annotated in this organism were these two, 0088 and 7579, which were truncated. Truncated means these had only the single stranded DNA binding site, and they did not have this acidic tail or the PG rich region. What is important is the acidic tail, because this is the acidic tail is where the DNA repair proteins bind. So if this SSB binds to the single stranded DNA which is coming up, and it is not able to recruit the DNA repair proteins, then the DNA repair cannot take place. So it will remain bound. So what happens if it will remain bound is, when I give it radiation, as we see in case of SSB1 overexpressing cells, that is this violet one, you see they have died, they are unable to recover. That is because it is only covering up the single stranded DNA but not bringing the repair protein. But if I did that with SSB2, I find that the yellow and green, yellow is a vector control strain and green is the one overexpressing SSB2, it is almost similar. And the reason for this was that though it was able to bind single stranded DNA, it was only binding those single stranded DNA which was part of a secondary structure. Okay, that which I did by using M13 kind of DNA. So then we started looking for that there should be a full length single stranded DNA binding protein, and based on the presence of the OB fold, which is the single stranded DNA binding site, and acidic tail, we found that one of the hypothetical proteins, ALL4779, actually has all this. Then we did biochemical studies and proved that it is a single stranded DNA binding protein with enzyme characteristics very similar to that of E. coli. And when we overexpressed this protein, yes, we found that the radiation resistance was actually much higher. Okay, so that kind of gave us this thing that yes, this is the single stranded DNA binding protein, which is the actual full length protein and is involved in DNA repair. Then in an organism which does not have reg BCD pathway, it should have the suppressors of reg BC proteins. So we started looking at SBCC and SBCD. Now these two proteins are usually found to function together. That is, unless they form a complex, they are not capable of repairing DNA. But what we found was we overexpressed these individually, and when we subjected them to radiation, you see here, these are like wild type or the vector control cells. After 7 days of recovery also, the cells have not come up properly. Whereas the SBCC or SBCD overexpressing cells, they have repaired themselves and they look as healthy as the control cells. So here we got a clue that yes, in this particular organism, the SBCC and SBCD proteins are actually functioning in a separate manner. They do not require to form a complex possibly. Since we were not able to purify this protein, we were not able to actually invocally prove that these do or do not form the complex. So this I will not go through much of it. So basically now we were concentrating on the RECF mediated HR, the homologous recombination ESD-SA pathway, as being the major pathway for repair. So this is the way it would be. So we were able to predict based on this as to which are the proteins which could be involved, and we have started the work on the RECF-OR proteins and the RECQ proteins right now. And why RECQ proteins we got interested in is, unlike bacteria, which has only one RECQ protein, in say 90% of bacteria, with the remaining 10% having two RECQ helicases, this bacteria has multiple RECQ helicases. And this multiple RECQ helicases is something which is very common in eukaryotes. Like in plants, it is 5 to 7. In humans, you have 5 of them. That is because they are all involved in different kind of functions in terms of binding to the kind of DNA structures they are binding to. So we tried to see why it is so. So here when we look at the RECQ helicases, we see that the 3 domains which are common to all the 3 RECQ helicases. For the Anabina ones, I have given it as AN as the prefix here. Okay, so we have the helicase ATP binding domain, C domain, and the zinc binding domain is common to all 3. And if we look across the organisms, the smallest of these Anabina RECQ was similar to the smaller REC-S protein of Bacillus. This REC-S protein is not required for normal cell viability, but it was required for stress response or stress resistance in case of Bacillus. And in case of RECQ1, in addition to the RECQ zinc binding domain, it also had the RECQ-C or the winged helix domain, and it had the HRDC domain, which is typical of what you see in E. coli and other bacteria. And it had an additional HTH domain, which could also contribute to different DNA structures which it could bind to. Then the third one which we had is RECQ2, which had these 3 domains, HTH domain and a priribosyl transferase domain. Presence of priribosyl transferase domain in a RECQ-like protein was observed for the first time in Mycobacterium. So, and these proteins were called as the RECQ-LH protein. So now this RECQ-LH protein is present in several pathogenic bacteria, and Cyanobacteria is not pathogenic. Okay, so now we feel that why did it have all these? So the work has just started, but we just did some kind of a phylogenetic analysis to see if they actually grew up together or they were 3 independent genes which came up. But what we see here is that all the 3 RECQ proteins of Nostoc belong to the same clade, and of these the RECQ1, which is very similar to E. coli, is actually coming with all other bacterial RECQ helicases, while the RECQ2, which has the priribosyl, comes with all the RECQ-LH type of proteins. And the smallest of the RECQ is the one which is also found in bacteria, in fungi and algae. And when we looked at human as well as in case of plants, we found that the RECQ1 is the one which is similar to Werner, which is what is you see even in E. coli, while the other 2 RECQ proteins seem to be RECQ, especially the smaller one, is the one which is similar to RECQ-L4 in human. So possibly there is some kind of a evolution of these RECQ, and in the bacteria, because they did not require multiplicity of these RECQ helicases, these were lost. But in complex organisms like humans or in plants, since the type of DNA structures which are formed are much more in number, these were required to ensure good translation, not just repair policy, but even during replication, these proteins are required. Okay, and then we just did bioinformatics study, a string analysis to find out which are the proteins they interact with, and we kind of found out that there are only a few proteins which are common for all the 3, but there are several proteins which are unique in terms of predicted interacting with these specific RECQ helicases. So possibly they are involved in different pathways. So since the work has just started right now, so we are still not identified the exact function of these genes. Then coming to oxidative stress response. The reason I'm putting it up is because we found something new in this particular response, that is when we talk about oxidative stress response means there are a lot of RO species. So what are the enzymes which are required is you need superoxide dismutase, catalase, peroxidotoxins. Now this organism has several peroxidotoxins, one catalase, and that is manganese dependent catalase, unlike in other organisms which have iron catalases. And here SOD, superoxide dismutase, it has both iron and manganese. But that is also not surprising because you could have any of these, copper, zinc, nickel, Mn or Fe. But what was interesting in this organism is that when we looked for the presence of this protein, we found that the MnSOD was present both in the cytosol and in the membrane. Now in bacteria, SOD proteins are cytosolic, but in plants and higher organisms, they are membrane bound. So this organism seems to have it at both the places. Our group, when we started working on it, Zhang's group had already published it, and they said that it is a 30 kilodalton protein where you see higher oligomers, higher sizes bands, because it being a membrane bound protein, the lipid is attaching to it, and that's why you get higher sizes. But we did a lipase treatment and we found that no, those sizes don't disappear. So luckily for us, what happened that we were not able to overexpress this protein in E. coli. But when we removed this hydrophobic region, which was 60 amino acids, then we were able to overexpress just the catalytic unit, which is much longer. I've just shown a few amino acids here. So that was about 24 kilodalton. Okay. So when we ran it with the total cell extract of 7120, we found that actually the lowermost band of the anabina extract ran with a 24 kilodalton of the protein which we were overexpressing in E. coli. So we knew that some kind of splicing is taking place. Not only splicing, but it is getting distributed in the cytosol as well as in the membrane. So that is where what we started doing is we made several mutants to prevent cleavage of the signal peptide, cleavage of the linker peptide, etc. And with these mutational analysis, we found that these cleavage of signal peptide and that of the linker were independent of each other. Okay, and we were able to identify the sites of cleavage. Okay, so I'm not going into the details, but we were able to unequivocally prove that the 30 kilodalton is spliced, the signal peptide is removed to give the 27 kilodalton, the linker peptide is removed to give the 24 kilodalton. And when we look at the zymogram, where you will see the active protein is usually a dimer, not usually, it is a dimer. And what we found were 5 bands here. So how do we get 5 bands? We have 3 bands here. So if they can form a dimer, you should have 8 of them, including the cytosolic FeSOD. Okay, but we found 5. So through a lot of analysis, what we found was a 30 kilodalton protein does not form the dimer. And what we see, band 1 is the 27 kilodalton homodimer, band 2 is a hetero of the 27 and 24, band 3 is a homodimer of 24 kilodalton, while band 4 is the FeSOD and 24 kilodalton homodimer, and band 5 was the FeSOD homodimer which was there. So we were able to actually show what the different forms actually were present. But the next question was, how do you get it in the membrane and in the cytosol? So what we predicted was that once the protein is bound, it is anchored in the membrane. So now 2 events can take place. One is that when it is anchored, the signal peptidase cleaves it off, so the 27 kilodalton protein falls off into the cytosol, and there it is cleaved further, so you get 24, so you get all the 5 forms along with the FeSOD. But it is also possible that after getting anchored, it gets translocated into the membrane. So once it gets translocated there, again the signal peptide gets cleaved. So this goes into the lumen area, the 27 kilodalton. So you have 27 and 24, and that is why you see only 3 forms in the membrane complex, the dimeric forms. So only bioinformatically we were able to show that there are signal peptidases which are capable of cleaving both in the cytosol as well as in the lumen region, and also the arginine C proteases also, which could be present in either of the regions. So that is how we said that this is a possibility, and this is the first time ever it was demonstrated that a bacterial protein can undergo cleavage so as to give a cytosolic and a membrane form. And but this was not news in case of plants, where in chloroplast they have shown it is present both in the lumen of the chloroplast as well as in the cytosol of the chloroplast. So but they did not give the concept behind it, and they have quoted our paper later on to show that this might be the possibility even in case of the plant chloroplast. So then we did some phylogeny just to see that whether there was the birth of the cytosolic SODs in bacteria took place during the evolution, and we just tried to compare the unicellular cyanobacteria, which are non-nitrogen fixing, and the nitrogen fixing filamentous ones. And what we found that the membrane bound MNSOD was present only in the nitrogen fixing bacteria. It was lost in the non-nitrogen fixers, and thus it percolated down, and the modern day bacteria did not have the membrane bound MNSOD anymore. Then coming to heavy metal stress, here my interest is not in the efflux proteins or how it is tolerating the metal stress as such, but what I was interested is in how cyanobacteria forms biofilms to protect itself from heavy metals. Why biofilms? Because biofilms are the ones where there is a lot of EPS formation, and EPS has a lot of, these are polysaccharides, and they have a lot of functional groups which are capable of binding heavy metals, all kinds of metals. And so here you see we demonstrated that even axenic cultures could form biofilms. This was Nostoc muscarum, and see we have treated it with 100 ppm of cadmium, and the cells were still alive, so they did not lyse, and these were only biofilms, not the cell suspension. And but you see here it's a gooey mass, and this gooey mass was nothing but the amount of EPS at which it was synthesizing. And here you can see with the AFM that actually this is without cadmium, and with cadmium see the amount of EPS which has got synthesized and resulting in an increase in cell size. And through FTIR and XPS analysis we were able to identify the functional groups to which of the EPS to which the metal bound. And it not only tolerated one metal at a time, but any number of metals. Here we have tried 3 metals, and all 3 metals could bind to it. So that means it had enough space for the metals. And what we also found that if you kept giving it more and more metal, the more and more functional groups were appearing on it, on the biofilm surface. As a result, it was able to take up the newer amount of heavy metals you were exposing. So we tried to look at what the EPS biosynthesis genes were, and this is kind of just based on small bioinformatics search which I carried out during the lockdown. So we found that they were actually present in, most of them were present in 8 clusters, and which I have stated here, and these are only gene names because most of them are not annotated. Okay, what interesting, we found that some of these, the non-clustered ones which we found, were the ones which were, some of them which were capable of introducing negative charge onto EPS. So we were able to identify some such genes also. But what we found interesting was the presence of glycosyl transferases, about 81 of them in number. And these are the ones which are annotated. And then when we were looking at some of the hypothetical genes, we found that at least 20 to 25 of them also had the same moiety. Okay, so why did this organism have so many glycosyl transferases? And if you look at this thing, they are actually generated from 2 glycosyl transferases that it is. So we are trying to see if there is a functional significance in case of cyanobacteria, because only 5 bacteria till now are known to possess more than 30 glycosyl transferases. Okay, and what we found is right now we have only characterized 2 of these genes. Both of them are responsive to cadmium, but they express at different times of exposure. So possibly this is to take care of the length of the exposure which might be taking place. So when we have talked about all these things, what is interesting is how do these genes get regulated? One I had already talked about was the heat shock response, but here I'm trying to look at either the global regulators or newer forms of regulation which could be present in cyanobacteria. The very first I'm going to talk to you about is a global regulator is Lexay. Now you will be surprised that why do I call Lexay as global regulator, because all of you would have heard of it as a SOS response regulator, which is a autoproteolytically cleavable protein in general. This is what is known in E. coli. it cleaves at between ALA and glycine because of a nucleophilic action of the serine residue. And RECA actually mediates it by binding to the Lexay protein, converting it from the non-cleavable form to cleavable form. So this is what is known in E. coli, and it was always thought that this is a SOS response protein. Now what was shown by Lee et al.'s group in 2010, that Cyanobacteria also has Lexay, but some of the Cyanobacteria have the non-cleavable form, but most of them have a cleavable form. So we tried to look in our own organism. We found that it was cleavable. As you can see, the two proteins here, this is the full length and this is one of the cleaved product, the other one gets degraded. But what we found that we could get the cleavable form if we are increasing the pH. That means we are providing nucleophile externally. But when we give it activated RECA, because I showed you there that in E. coli it requires activated RECA, which changes the conformation of the Lexay protein and then cleaves. But here if I give it activated RECA, it did not cleave the protein. That means it has a RECA independent mediated cleavage here, only alkali. So what kind of an environment occurs inside Cyanobacteria to allow its cleavage, we still don't know, but the cleavage does occur. So if that is so, in case of Synechocystis, which has a non-cleavable Lexay, they showed that it is not involved in DNA damage response, but involved in carbon metabolism. Now here we have in Anabina a protein which is cleavable, but not classical cleavage is there. So what we did is we tried to see its response when we overexpress this protein. What is its response to many stresses? So here see carbon metabolism, when we inhibit photosynthesis, the stress response is better. Like these are the cells which are overexpressing it. So these are much better than the ones where it is not overexpressed. But if you give it radiation, then here you see this is not as good as the control cells. Same way in case of oxidative stress, the top and the lower one you can see here, and metal stress. That means for all other stresses it is actually becoming bad. So why is it so? So we tried to look at the proteomic approach, and what we found was that some of the proteins are actually down accumulated and some are up accumulated. Now in E. coli we knew it was only a repressor, but Synechocystis, the unicellular cyanobacteria, they showed it is a actually an activator. And here we see a process where it could be both. So we looked at the genes, and we tried to see by EMSA studies, promoter analysis in E. coli, where we are overexpressing the Lexay, and we were able to identify 17 genes which are regulated by this gene, 3 of which were found to be upregulated. So now the next thing was, okay, this I won't go to too much detail. This is basically to show that Lexay actually now plays a global regulatory role because it is affecting photosynthesis, so carbon metabolism also. Photosynthesis, it doesn't change much, enhances carbon fixation, decreases carbon catabolism, so enhances tolerance to photosynthetic inhibitors. But since it brings down the levels of the oxidative stress elevation proteins and the DNA repair proteins, it causes cell death in the presence of oxidative stress and any stress which can induce DNA damage. But what we were interested was that at that time there were 3 or 4 other groups which were looking at Lexay mediated regulation in Anabina. But each one of them was working on 1 or 2 genes, and depending on the gene they were predicting what is the Lexay binding site. Now you cannot have a global regulator which will bind to just some region depending on the gene. So you need to have a consensus sequence where it is binding, and that it binds as a dimer is something which we had been able to show. So we just did, we matched the region. We had narrowed down to about 50-60 bases where the binding occurs. So we had all the 17 genes and we looked at it, and we found the palindrome which is AGT, ACT, and the spacing was between 4 to 11 bases. Plus there were certain cases like here you see AGA and ACT, but this happened only if it was part of at least a 4-base palindrome. You can see here as well as here. So we gave it that it could be AGT, ACT with the possibility of 1 base mismatch if it is a part of 4-base palindrome. And then we tried to prove that yes, these arms are important by mutating one of the arms and showing that the Lexay does not bind. At the same time, we decreased the spacer region from 8 to 4, and we showed that it is able to bind. So which was again a little surprising, because if it's a dimer, it cannot have so much flexibility in the spacer region between 4 and 11. So the reason was that the Lexay protein had a flexible region which allowed it to bind from major groove to major groove of the DNA, as well as major to minor groove of the DNA. And this is based on predicted structure, not the actual structure. So we were able to prove that this could be the thing. And based on, then we did, I won't call it a genome-wide study, but just some clusters I looked at, and we found that in these clusters, I was able to identify these many genes which had the AN Lexay box. And then subsequent studies, I was able to identify that in photosynthetic PETC genes, 70 of the 92 genes had the Lexay box. And similarly, nucleotide synthesis genes, radiation responsive genes, and cadmium stress genes also. Then with further studies, actually I was able to narrow down onto the binding site and showed that if there is a mismatch and you have ACT in the right arm, these were the only things which were allowed in the left arm, not the other mismatches. Similarly, if AGT is in the left arm, these were the nucleotides which were allowed in the right arm. At the same time, though the binding is not affected, the regulation by Lexay was achieved only if it was within 100 bases of the promoter region. So these are the things which, because I was able to do transcriptional studies in these cases, I was able to identify that much. So this was the other, when we were discovering the global regulatory role of Lexay, the NTCA's global regulator, NTCA Pipex, Pipex is a co-binding protein with NTCA. They were Episonas groups. They showed that this is also a global regulator. As you see here, it is involved in nitrogen metabolism, which is known worldwide, and also in several hypothetical, in translational protein, cell cycle control, other regulators, stress metabolism, potassium transport, etc. So this, but one thing which was not known was of DNA repair proteins. So we tried to see if that happens for this thing. So we looked at just the REC-FOR genes which my PhD students started working on, and we just looked at the transcript levels under nitrogen fixing and nitrogen supplemented conditions, so the yellow bar, and we see that under nitrogen fixing conditions they were high, which indicated that NTCA may be playing a role. So we did identify the NTCA box, which is this yellow color, and when we overexpress the NTCA protein in E. coli, we found that the promoter activity actually increased. That showed that yes, NTCA is involved even in the regulation of some of the DNA repair genes at least. Okay. Now, as you see here, for all these genes, we found NTCA box, then we found the Lexay box, which is red and pink. That is because DNA repair genes are regulated by Lexay, and here you can see the downregulation, the red one. But what is interesting is the presence of these blue boxes. These are heptamer repeats. Okay, so these were things which we found, and we deleted these heptamer repeats also to find out if it has any role. So the one which we found upstream of the RECF gene is this kind of repeat, TTC AAA, which was found in 3 consecutive repeats were found. And what we found was that this was responsible for the accessibility of the promoter for transcription, because this one overlapped with one of the promoters, which bioinformatically is predicted to be a very strong promoter. But in vivo, this promoter was not accessible unless we deleted the heptamer repeat. Okay. And second thing which we found was that it was also possibly controlling the translation, because this particular gene had a non-canonical SD sequence, and which was very far from the ATG, which is normally it has to be 4 to 8 bases, and in this case it was around 12 bases. So possibly this may be having a role even in that, but we were not able to prove that. So when we looked at in the genome, we found that this was present in at least 21 genes within 100 bases of the -10 region, and in most of these, excepting 2, the SD sequence was non-canonical. Okay, so that kind of gave us this thing that possibly this may be true. And it was not linked to Lexay or NTCA based regulation, because not all these genes had NTCA and Lexay regulation. Similarly, for the REC-O gene, what we found is this kind of a repeat, GACAAT or GATAT. I think I've forgotten one C is also there, GAC. Okay, because it's a heptamer, GACCAAT. Okay, so what we found was, no, it's okay, sorry. So what we found was that when we deleted this particular box, we were able to get regulation, negative regulation by Lexay or positive regulation by NTCA in E. coli. Whereas in case of Nostoc or Anabina, it was able to regulate. That means there might be a protein which is able to recognize this heptamer, resulting in that being removed. As a result, you are able to regulate through NTCA and Lexay. Secondly, the SD sequence was present within this box. As a result, it may be involved in the accessibility of the SD sequence. Again, this was present in 14 of these 14 genes, and none of these were like, it was, again they had only 2 of them had canonical SD sequence, and most of the time it was very close to the translational start site. Okay, so that means these are involved in regulation. So this is a very basic search where I have looked at it, but I have a feeling that these heptamer repeats, I have found one more upstream of a Eps biosynthesis genes, which I have not done any further studies. But what I feel is that these heptamers may be in plenty in this organism, and there is a need to look for it, because till date there are only 2 publications relating to the role of heptamers, where they have predicted, one in Beckettosi and the other in I think in Saccharomyces, where they have shown that the heptamer is required for DNA binding or for translational control, and that is also purely based on prediction. The other interesting thing which we found was actually was a rare initiation codon which we identified, that is through the Rekar gene. And how we did it is, the annotated Rekar gene was very small, it was only 13 kilodalton protein, and what it lacked was this 4-cis zinc binding domain, which is essential for Rekar function. So this was absent in this organism. So first in the annotated Rekar. So first we looked for a full length Rekar in the genome and we didn't find it. So we started reading upstream of this particular gene, just translated it, allowing it to take any codon as the start codon, and then compared it with the known Rekar sequence in other bacteria, and we came across that this GTT could be the possible start codon. Till date, only in for one particular gene, that too in Saccharomyces, they have shown GTT as an initiation codon, but that is very poorly expressed gene and a non-essential gene. Whereas Rekar is an essential gene because it's part of the DNA recombination repair pathway. Okay, and then when we saw this, we found that this GTT is present in several bacteria, and in all of these, several cyanobacteria, and in all of these it was wrongly annotated. Okay, so how will it function as a can it function as a translational start codon? First of all, it had one good thing was that it had a canonical SD sequence, though a little far away, but it was a good SD sequence. So then we looked at the FMET tRNA and we found the complementary sequence of GTT within the anticodon loop of the FMET tRNA in case of Nostoc. So this GTT, though a rare codon, rare initiation codon, could be one of the initiation codons in the Cyanobacterium, and there may be a need to re-annotate several other genes where this must have been missed out. GTT as an initiation codon, because normally what initiation codons we take is either ATG or NTG to some extent, any of the GTG, etc., CTG, TTG. So this GTT as an initiation codon is something which we should take up and try to see if there are many genes which have this. So now I will not be concluding my talk, but I'm just trying to put forward my thoughts to it. What I feel is that as you saw during my presentation, there's a lot of multiplicity of genes in this organism. This could have helped it to combat the harsh environmental conditions, and with the evolution of microbes in a closed niche, this multiplicity was possibly lost because they are now getting exposed to very small number of stresses, so possibly this multiplicity is not required. But with increasing genome complexity of higher eukaryotes, the multiplicity was retained and also enhanced further. Second thing is that what we see is a multifaceted regulation of genes, as I showed for Echefoan, and presence of global regulators allowed cyanobacteria to adapt to multiple environmental stresses. Now, there possibly now down the line, evolutionarily advantageous modes of regulation only were retained by microbes and eukaryotes. Okay, this is a small group. We are a fun-loving small group which works together. And thank you all for listening. So I'm open for questions now.
Questions?
Professor Bhaskar, I have a very general question.
Yes, sure, sir.
See, you talked about different stresses, oxidative and heat shock and so on. Suppose a bacteria is exposed to 3 stresses simultaneously.
Yeah.
Do they have a different strategy altogether?
No, actually, see, most of the bacteria die. Die.
Anything more than one, I see.
Yeah. But here in Cyanobacteria, I have myself done multiple stresses, 1 or 2 stresses, like salt and heat together, osmotic and heat together. And we've also accidentally done heat and radiation together. The survival does go down in certain cases, depending on which treatment it goes. But because of this global regulatory, because of the global regulators which are present, it is in addition to synthesizing just proteins which are required for heat shock, it is also able to synthesize other proteins.
I see. There is something common.
There is common. That is why global regulators were there, because for Cyanobacteria it was never that it was exposed only to radiation when it came to existence. There was heat also. So it had to develop that it cannot have the gene with different regulatory elements. So instead it had a common regulator which was able to regulate all these genes. So whatever was required, it was able to synthesize. So even we found some of the heat shock genes also regulated by Lexi.
Thank you.
So that we have been able to do. Yeah.
Hi, nice talk. I had one quick question about the evolution part in the, so you think there was a multiplicity of genes that was in ancient cyanobacteria and got lost but got retained in modern eukaryotes. So I think the understanding is that eukaryotes probably evolved from archaea via endosymbiosis or something. So
yeah, that is what is said. See, for plants it is Cyanobacteria which is thought to be, because a lot of chloroplastic genome what you see in chloroplast is present in Cyanobacteria already. So there is the thing that it would have evolved in that way. So there could be a lateral transfer or a horizontal transfer of these genes. So when I'm talking about it, I'm not saying just in case of this thing. And since you mentioned archaea, we do see that many of these Cyanobacteria and archaea genes are similar. And between the two, who came first is something which is very difficult to say. But if you just look at the radiation response, which is the first stress which was there, and the dating, the Cyanobacteria feature has been found. The fossil data shows Cyanobacteria are older than archaea. Yeah. So whether they developed later on, that is what is not known. So what I am not since I have not studied how to look at the evolution as such, but this is a very basic study which I have done to look at it.
And another quick question about the motif analysis. So in particular that dimeric motif AG, I think it was ACT dot AGT dot with the variable length.
Yeah.
It looks like a fairly non-specific motif that would also occur randomly in a lot of the genome.
Yes.
So you did a control of is it overrepresented in these genes versus?
Yes. See, what I was looking for is only, see, this kind of a motif can be present across. But where would it be actually available for binding? Not when it is within the gene. So in the DNA, so I'm trying to look at where it is going to bind and be responsible for regulation. That can be only in the promoter region. Okay, so this motif, we were able to prove that this motif is the one which is responsible for Lexi binding. But we were not able to point out as a bigger motif, because in case of E. coli also, it is 6 base pair motif which is given, but again on that 3 only are conserved. So this is a slightly non-specific. Yes, I agree that it could be present across the genome, but involved in regulation only when it is present in the promoter region within 100 bases upstream or downstream, but not within the gene. Within the gene, it is not regulatory in nature.
Okay, thank you.
Questions?
And the other thing is Lexia levels are so low in this organism that it cannot keep binding to these regions.
Hello, ma'am.
Sure.
Thanks for the nice talk.
Thank you.
So I was wondering, just a naive question because I'm not from this area. So when you measure, when you do the sequencing, so how do you identify the genes in this complicated organisms? So
No, see, identification of genes, I mean, the genome data was not done by me. It was in 2001, where they annotated based on a normal ORF finder kind of a thing, where you take any of the NTGs, ATG, GTG, any of the NTGs as a start codon, and you look as you normally do. And then they try to annotate based on the functional groups which are present, either the domains or these things, and by what is known in E. coli, Bacillus, Salmonella, etc. And in some cases with higher plants, where it has been annotated. That's how they were able to annotate this. But when I have to do it, means where I found, here I have shown only RECAR, but we re-annotated NDK, which is nucleotide diphosphate kinase also. There also we found that the protein which is annotated, the gene which is annotated is coding for a smaller protein, and we were able to annotate the actual starting codon also. So that is done manually, and by making some constructs to show that this is the one which is being expressed.
but sometimes there are these misses, right? Sometimes it doesn't, means the genes don't give the protein because there's maybe some failure in the process or
Yes,
some
yes.
copying
See, not
failure.
all genes are going to code for proteins and give rise to proteins, because there may be, while reading through, they may not, there may be secondary structures in the RNA which will prevent the translation, or there could be some stop codons which are getting introduced during this thing. So there is a possibility of that. So these are protein coding genes only based on annotation, that these look like protein, they are not coding for RNA, because RNA genes were known. But since the ones which I am saying is hypothetical and unknown are because similar proteins have not been shown for other organisms. So that is why people say that they are not unknown, but where you get similar proteins, you can at least give an annotation to that.
Thank you.
Questions?
Sure.
See, mammals are affected by viruses.
Yeah.
So do the bacteria also have
Yeah.
viral stress, and how do they manage?
Okay, that's a good question. So you have heard of bacteriophages?
No.
Bacteria, they are called as phages, they're not called as viruses per se. So bacteriophages are the ones which can get into bacteria and kill them also. But they have a system to overcome that, because by preventing the cell lysis, the way we humans respond, the bacteria also can respond. And same way, they don't need immune system. See, we talk everything in terms of immune system is to destroy like shoot a gun at the virus. Here what it does is, see, any virus has two phases. One is the lytic phase and the other is lysogenic phase. Lytic phase is when it is going to divide and get out of the cell. So that is when it kills. But when it is in the lysogenic phase, it is only multiplying. It is not going to disturb the system. Okay, so the bacteria will have processes to prevent lytic conversion of lysogenic to lytic phase. So if it is able to do, the bacteria survives. If it doesn't, it dies. So even for cyanobacteria, we have cyanophages, and many of these cyanophages are also found near the blooms and all. It can be, it is not for humans. It is not pathogenic for humans, but for plants and algae, it can be pathogenic. So that is how it is. And now bacteriophages are being used for treatment also now. So.
Questions?
Okay. Hi, is there any specific reason that you looked at the biofilm formation?
Okay, the reason was, it was more of an accidental discovery in my case. Because biofilms are known for metal tolerance as such. My interest came was that till then, most people were talking about requirement of more than one organism for biofilm formation, especially with respect to cyanobacteria. Okay, so since I found it from an axinic culture, that if I kept it stationary with slightly lower light, I was able to get biofilms. So I started looking at these biofilms, and slowly discovered that it is making lot of exopolysaccharides, and now my whole attention is going to exopolysaccharides and pathways. I'm also looking at how I can improve the quality and quantity of this polysaccharides, because the metal bioremediation is very important. This is actually giving me a chance to develop a green technology where I don't have to go for chemical means of bioremediation. I'm using an organism which is photosynthetic in nature, so I need only water, air and sunlight. So I'm not going to get into any kind of chemical synthesis. So environmentally positive organism I will be using for this purpose. So that is my this.
Questions? Okay, then I have a few.
Sure.
So the first question is that typically E. coli and Bacillus, they have about 4,000 genes. You said Nostoc has about 6,000 genes.
6,000.
So it's about 25% larger.
Yeah.
But when I look at the number of sigma factors, E. coli has 7, Bacillus has 14, and you mentioned 12 here. Do you
12
have
identified.
12 identified. So there could be more?
There could be more, because we have not identified from the hypothetical ones.
Okay, that was one. The second question would be that this increase in the genome size with respect to E. coli and Bacillus,
I would call it decrease.
Would we decrease?
Yeah, because decrease from Cyanobacteria to E. coli.
Okay,
because E.
decrease
coli is
from-
modern.
Okay, so what is new? What is extra? Is it largely to photosynthetic machinery or something else?
No, it's not because of photosynthetic machinery only. That accounts for only 92 genes.
Okay.
Okay, and this is more because many of these genes, like I showed you HSP60, there are 2. DNA-K or HSP70, there are 7 of them. And SOD, there are 2 of these. Then like glycosyl transferase, I've shown 81. So like this, these are the ones which I have worked with. But there are several, and helicases, DNA helicases, I've shown 3 RECU helicases, 1 RECG is there, and there are 25 DNA helicases in this. So these are the way it has actually expanded, and then it has been lost over the evolution. Even among Cyanobacteria, it is lost.
So you showed that this Nostoc is highly tolerant to radiation.
Yeah.
So do you expect the ROS metabolism to be highly evolved here?
Yes, it means it is, it has all the genes which are required in terms of taking care of superoxide, then H2O2, as well as peroxides. And for peroxides, which is the most common one, superoxide is more because you have oxygen evolution, so you're going to get superoxide. That is why you have the SOD even in the lumen, thylakoid lumen, to take care of the oxygen ROS which is being generated there. And it has about 7 or 9 peroxidotoxins which are capable of taking, and each one of them is indispensable. Means you can knock down some of them, but you cannot knock them out. So each one of them are contributing to some extent, all of which would be expressed under certain conditions and taking care of it.
The last question would be that, so which organism should one focus on, or what should be the strategy to improve functional genomics in general of Cyanobacteria? It seems to be a general problem you're facing, right?
See, the problem we have is that we cannot overexpress. I mean, if you were looking at functional this thing, we are not able to overexpress all of these in E. coli. Now we have developed the system to overexpress these proteins in Anabaena itself. So as to, I mean, not overexpress just for physiological studies, but even for purification, using his-tag purification kind of things. Because most of the protein synthesis is very low in this organism. Maybe because of the multitude of genes. You don't see proteins except the photosynthetic or the pigment proteins which are seen in bulk, or some of the heat shock proteins. You don't see other proteins in amounts that you can isolate it by just growing the cells to larger volumes. You can't do that. So right now we are trying his-tag purification in this organism wherever it does not fold in any of the E. coli strains.
Thank you very much. Any further questions from anyone? If not, let's thank the speaker again for this nice talk. Thank you, ma'am, for the talk. We hope to have you again soon.
Yeah, thanks a lot to all of you also.